Hello, this is Dr. Shurpneni from United Medical Group. Topic for today is aging. What is aging? Aging can be thought of as both biologic and can also be thought of as social. One ages with time, but the physical manifestation of aging is directly related to the biological process of your cells. When your cells lose their native function due to stress, oxidative stress, chemical stress, stress imposed by injury, diseases, you age. According to World Health Organization, the number of people ages 60 and above by 2050 will almost double from 12% to 22%. By 2050, 80% of older people will be living in low and middle income countries. The pace of population aging is much faster than in the past. Aging is not only going to be a health issue, but also a socioeconomic issue. Unfortunately, age has an inverse relationship on our cognitive and physical function. As a result, our productivity gradually declines with age. What is aging? Quick definitions. A genome would be the entire composite of genes that you comprise in your cells. And your genome is divided into 23 pairs of chromosomes. These chromosomes are essentially packaging material for DNA. DNA needs to be carried and stored and transferred, copied. So chromosomes essentially help keep DNA organized with the help of proteins called histone. DNA, which is a double-stranded molecule which connects with nucleotides and forms the classic shape of a chromosome, the double helix structure. DNA itself is comprised of nucleotides called adenine, thymine, guanine, cytosine, which form a unique sequence to eventually give rise to physical characteristics or functions to sustain life. Think about your hair color, your eye color. This is represented by a genetic sequence which is very unique to that characteristic. Essentially, nucleotides which can be subdivided into purines and pyrimidines can form bonds with their opposing base pair. So A connects with T and G connects with C. If you know the DNA pattern of one strand, you can predict the opposing pattern because you know the connecting pair. DNA is what makes you who you are. Interestingly, the coding material, which are the genes, about 20,000 genes in the entire human genome, represent 1% of your DNA, and the rest, the sequences, represent proteins, non-coding DNA, introns, which ultimately interact with your coding DNA to help form proteins, which are essential for life. But what is epigenome? According to the National Institute of Health, epigenome is a multitude of chemical compounds that can tell the genome what to do. The human genome is the complete assembly of DNA, about 3 billion base pairs that makes each individual unique. DNA holds the instructions for building the proteins that carry out a variety of functions in a cell. Epigenome is made up of chemical compounds and proteins that can attach to DNA and direct such actions as turning genes on or off, controlling the production of proteins in particular cells. Are you feeling confused? For instance, think of cells that perform certain activities. Take, for example, the hemoglobin, which carries oxygen in your blood. Now, this function is directed by the epigenome in the cells in your bloodstream, which direct it to make protein like hemoglobin. You carry the same genome in all of your cells, but the genome in your muscle tissue, bones, blood have to act differently to perform tasks that are more specific to their host cells, like the muscle cells or the blood cells. So the epigenome controls which genes are turned on or turned off in these cells. An example of such influence on DNA is a chemical modification process called DNA methylation histone modification. Methyl groups are attached or tagged to the DNA molecule, which directly affects the function of this DNA by telling the genes to turn on or turn off. The other process is histone modification, where DNA is essentially neatly packaged or wound up with the help of this histone protein, which acts like a spool-like structure to keep it neatly organized and packaged in your nucleus. Proteins can easily access histone and attach chemical tags, which determine if a part of the DNA could be accessed or it's not available. This is a representation of the histone modification process. As you can see here, the chromosome in a nucleus, which contains DNA, is neatly packaged or organized with the help of the histone protein, which acts like a spool, shows tiny little tags like the chemical tags, which represent areas where DNA can be accessed or it's inaccessible. What you truly inherit is your genome from your parents, but the epigenome is relatively unique to each individual generation of offspring. Your epigenome starts over, so to speak, but a part of the epigenome from the previous generation, from the egg and the sperm, can be passed down. And when you inherit this partial epigenome from the parents, they replicate and divide in your body as your cells multiply and divide. That determines the individual function of these cells. This leads me to the topic of imprinting. Imprinting is a process where one gene is turned on and the other gene is turned off. All of your genes have two copies, one representing your mom, the other representing your dad. Some cells have genes from your mom that are turned on, and the other cells have genes from your dad that are turned on. Now, which are turned on and which are turned off is really determined by the epigenome, which functions as an augmenting layer of these genes. This epigenome, which allows our cells to specialize, can be altered or affected by stress, both mental and physical, diseases, and chemicals. So, for example, if you smoke tobacco, the chronic damage to the epigenome with exposure to the chemicals in your cigarettes can lead to our inability to control the genome in order to switch on or off genes that are protective to prevent diseases like cancer. This loss of function or malfunction of the epigenome is undoubtedly a factor in processes like cancer, diseases in the human body. Scientists looked at a cohort of 1,700 children in Great Britain who were considered socioeconomically disadvantaged. What they observed was they had genes that were distinctly affected with DNA methylation pattern that were vastly different from children who had a normal upbringing. And these genes that were affected had to do with functions like metabolism of air pollutants, processing tobacco smoke, and inflammation. This suggests that the environment has an effect on your epigenome and can alter how your genes respond. Currently, there are ongoing studies and research into understanding epigenomics, which I believe will unlock our ability to help manage disease, prevent disease, and certainly extend life.